good evening everyone. Welcome to the live session of week 5. So in the previous session we were discussing about MHC molecules, right? And we discussed how what is the structure of these MHC molecules, MHC class 1 and class 2, how do they bind to the antigenic peptide, and also what is the structure of the MHC co-receptors like CD4 and CD8. And in the previous session many were actually asking how does this antigen presentation by MHC molecules occur. So I tried to discuss a little bit in the last session, but today I will go in some details about how antigen presentation occurs in by the help of these MHC molecules. Now, before going to that, this is the structure of the MHC molecules, MHC class 1 and class 2, and so this we had discussed in our previous session also, that there's a peptide binding cleft. First of all, there are two chains. First of all, there are two chains, the alpha chain and the beta chain. There are two chains, the alpha chains and the beta chain, and there is a groove that forms between these alpha and beta chain domains, which actually serves as the peptide binding cleft of these kind of MHC molecules. Now, we also discussed in the previous session that MHC molecules are polymorphic, right? So this also we were discussing that polymorphic means it can have various alleles, okay? And actually in our population, in the human population, there are approximately thousands of MHC alleles present. Now, no other gene does not have these many alleles. So here I will introduce another term, which is polygyny, polymorphism and polygyny. So how are these two different and what is the role of these? So polymorphism basically means that you can have many alleles in the population. Now remember one thing, that polymorphism is considered in respect of the population, right? So it's like there are many alleles present in the population, many MHC alleles, but however in one single individual there will be only two chromosomes, right? So one single individual can have only two alleles as shown here, right? So therefore that does not generate much diversity of the MHC molecules. However, polygyny is one event which has taken place in the course of evolution. That is, these MHC genes have been duplicated. Duplication of these MHC genes have taken place, which is why you can now find many MHC genes. Here you can see that there are 3 MHC genes which you can see here. So usually we know that there will be a single gene for any protein product or for any function. There will be a single gene, but here in case of polygyny you can notice that MHC molecules are actually coded by multiple genes in any single individual. So this is individual based. So now you can understand that in this particular individual there will be two alleles, right? And also there will be multiple genes. So now if you like when these events take place together, polymorphism and polygyny, You can now see the picture carefully. You can see here that there are many genes. Not only that, these two alleles are very different. Okay, so the polymorphism and polygyny both equally contribute to the diversity of MHC molecules. Alright, now how does this diversity of these MHC molecules, what is the molecular mechanism behind it? So the mechanism is one thing you can actually assume, that is recombination, genetic recombination, right? Something that we had studied in case of antibody diversity, right? That genetic recombination creates diverse antibodies, or the variable diversity in the variable region of the antibody. And one interesting thing is that there is another event which takes place which contributes to the diversity of MHC molecules, which is gene conversion. So in this figure you can see here, so I was telling you this, that the ancestral MHC gene, it undergoes duplication. So that creates so many rounds of duplication and also divergence, creates multiple MHC genes in any individual, right? Next, what happens, there is one event which takes place, is gene conversion. What is gene conversion? Gene conversion occurs when, like, during the crossing over. So when crossing over takes place during meiosis, the homologous chromosomes are aligned to each other, right? We know that during meiosis the homologous chromosomes are aligned to each other, and there is another event which takes place here, which is crossing over, right? Through the chiasma formation and all that. So during this kind of event, during this crossing over event, what happens, these MHC genes, they also align, these chromosomes also align to each other, right? And while this kind of alignment takes place, you can see here clearly that if these chromosomes are misaligned, right, if some kind of misalignment takes place, so what happens, this yellow, here you can see here that this yellow part should actually align with this yellow part, but here that kind of event is not taking place. This yellow part is aligning with the green part. So green, yellow, what are these? These are different MHC genes, right? So these are aligning in a wrong manner, right? So this is not the correct alignment, but however this kind of alignment takes place because there are so many genes in tandem, right, in an array, which is why there is often this misalignment which takes place. And if there is a misalignment, then what happens, then recombination, like not recombination but crossing over will also take place, right, in this particular position. And if crossing over takes place, what you can see here, that a part of this yellow gene is transferred to this green part, right? So there is exchange of the genetic material between these two chromosomes, right, by crossing over. And during this time, what happens, that so after the chromosomes are separated, after meiosis, after the end of the meiosis, you can see that this green gene will now have a part of the yellow gene as well. Okay, so you can understand that this again creates another round of diversity of the MHC molecules, right? So polymorphism and polygyny, and together with gene conversion, so these are the events which determine how the MHC molecules will be very diverse in the population as well as in any individual. And remember, the particular combination of MHC alleles which are found on a single chromosome is known as MHC haplotype. Okay, so this set, suppose this is called an MHC haplotype. Okay, and one other thing that I want to mention here is that MHC molecules are also codominant. Can anyone explain what is codominant? What do you mean by codominant?
When I express both the alleles characters.
Yes. So there is nothing as like a recessive character or a dominant character. Both the alleles will be dominant here. So they will both be expressed. So which creates another round of diversity, because otherwise if one allele would be dominant, then only that type of MHC allele would be, MHC molecules will be produced, right? So MHC molecules are also codominant, and which is why it's very interesting that even like because of these many events which generate the diversity of the MHC molecules, uh, any two individual will not have the same MHC haplotype, and also because of that, even in between siblings, there is a huge difference that is found, which is why during if you have to do organ transplantation or tissue transplantation, then in that case this kind of rejection, the graft rejection, takes place, because the MHC molecules are not same in any two individuals, right? So now if you have understood this, I'll go to some questions here. Peptide binding cleft of class 2 MHC molecule is formed by which of the domains? Please let me know your answers in the chat box. Okay. Any other answer? Most of you are answering A, which is the correct solution here, right? So the class 2 MHC molecule, it is somewhat different than the class 1 MHC molecule, in which the in the class 2 MHC molecule, the beta chain has two domains, beta 1 and beta 2, and the peptide binding cleft is formed by alpha 1 and beta 1. So that is the right solution. Okay. And then another question: polymorphism of MHC class 1 and 2 occurs due to which of the events? Okay, most of you are answering D, which is both gene conversion and genetic recombination, which is the correct answer. Okay, not post-translational modification. Post-translational modification does not have a role in this kind of polymorphism. Okay, so now I'd like to go to the antigen presentation part of the MHC molecules. So in the last class we were discussing like how the MHC class 1 molecules present the antigen, where does it get its antigen, how is it expressed on the cell surface, and similarly for class 2, what happens for class 2, is it the same or is it different? So now we will answer this one by one. First of all, what happens during antigen presentation of MHC class 1? So what happens is that first you can see MHC class 1 is responsible for presentation of intracellular antigen, right? Kindly excuse my voice today. I'm actually down with viral fever, so I'm just speaking lightly here. Okay. So antigen presentation by MHC class 1, and MHC class 1 actually expresses intracellular antigen, right? And here you can see that when a virus infects the cell, so what happens, these viral proteins will be synthesized in the cytosol. So here you can see that the viral proteins are being synthesized by the ribosomes, right? And because of which what happens, now these proteins will now have to be bound to the MHC molecules, right, to be finally expressed at this cell surface, right? Now remember one thing, viral proteins are being synthesized in cytosol. However, when the MHC molecules have to present their present antigen, they will present it in the form of peptide fragments, right? So how does this fragmentation of the proteins take place? Okay. Now we are actually discussing this also in the last class. We talked about something called a proteasome. So this is known as ubiquitin proteasome system. Okay. Here what happens, so this is the proteasome system, right? So there's a core which is 20S, so this is a core, and there are two cap-like structures, two 19S molecules which form the cap-like structure, right? And what it does is that first the protein has to be, for any protein to be degraded, the protein has to be ubiquitinated. What is ubiquitination? So it's nothing but addition of ubiquitin, which is a 76 amino acid protein. Okay, so how like this is called ubiquitin because this was found ubiquitously in the cells, right? Which is why it was named as ubiquitin. And this is a 76 amino acid protein which is added to the protein molecule, the target protein, which has to be now degraded. Okay, so how does this take place? So lysine 48 of the first ubiquitin molecule is linked to the C-terminal glycine of the second ubiquitin molecule. So the first ubiquitin molecule is associated with the protein, the target protein, and then the lysine 48 of the first ubiquitin molecule, the lysine 48 amino acid, 48th number lysine, is now added to the C terminal of the glycine of the second ubiquitin molecule. Okay, so this is how ubiquitins are joined together. Okay, and remember, this is polyubiquitinylation. So many ubiquitin molecules will be actually added, and that has to be like more than 4. Okay, so at least 4 ubiquitin molecules will be added to the target protein, and then so these polyubiquitinated proteins are recognized by this 19S cap. Okay, and this actually carries this protein into the core of this proteasome. Okay, so this here you can see this protein, this has this ubiquitin tag, and it is being carried into the core of the proteasome, where the proteasome will degrade the protein and it will make peptide fragments. Okay, so this is how any proteins are degraded in the cytosol. And for example, like viral protein I was talking about, so these viral proteins will be degraded in the proteasome like this manner, and so that will generate peptide fragments. Now the important thing is that actually this event takes place in the cytosol, because the polyubiquitin the ubiquitin proteasome system is present in the cytosol. But now how these cytosolic proteins will move to the endoplasmic reticulum, because you might have seen here that this is actually the endoplasmic reticulum, right, where the MHC molecules bind to the antigenic peptides, right? So if the proteins, these are being synthesized in the cytosol, now these are degraded in the proteasome in the cytosol itself. Now how will they transfer? How will they be transferred to the endoplasmic reticulum? Because if it is not transported to the endoplasmic reticulum, it will not be bound by the MHC molecules. Clear? So now we will see that how does this take place, how are these peptides transported to the endoplasmic reticulum for binding to the MHC molecule. And here you can see, first of all, the MHC class 1 molecules, they are in the endoplasmic reticulum, so they do not exist in its properly folded state initially. So you can see here, this is a partly folded MHC class 1 molecule. Okay, partly folded, and also the alpha chain is present, but the beta 2 microglobulin is not yet associated with the MHC class 1 molecule. Okay? So this happens in the endoplasmic reticulum, the lumen of the endoplasmic reticulum. And here one protein molecule, which is a chaperone, which is named as calnexin. So calnexin is a chaperone which holds on to this MHC class 1 molecules, and it until the beta 2 microglobulin binds to it. Okay, so until beta 2 microglobulin binds, calnexin is present to maintain the MHC class 1 molecule in its partly folded state. Now next what happens, this beta 2 now will bind to the MHC class 2, MHC class 1. Okay, so this beta 2 microglobulin will now bind to the MHC class 1, and once that to make that happen, the calnexin is released from the MHC class 1. Okay, calnexin is, here you can see this calnexin is being released, and then the MHC class 1 molecule binds to the beta 2 microglobulin. However, another round of chaperones, another set of chaperones now come into action, and this is known as the peptide loading complex. Peptide loading complex is composed of your calreticulin, tapasin, and ERP57. So these are the proteins which form the peptide loading complex. So once calnexin is dissociated from MHC class 1, this peptide loading complex binds to the MHC class 1 molecule, and what it does is that, what is the function of this kind of peptide loading complex? Calreticulin is a chaperone, again like calnexin. Tapasin is tap associated protein. So here one protein molecule you can see, which is tap, and tap is actually a dimer of tap 1, tap 2. What is tap? Tap is nothing but transporters associated with antigen processing 1 and 2. Okay, transporters associated with antigen processing. The principal role of tap is that once these peptide molecules are generated in the cytoplasm, after the degradation by the proteasome system, these peptide molecules are transported into the ER lumen by the help of this tap. Okay, so tap is like a transporter which transports these short fragments of peptides into the ER lumen so that it can now bind to the MHC class 1 molecule. And tapacin is nothing but the tap associated protein. It does what? So it actually creates a joining, like it creates a connection between the MHC class 2 MHC class 1 and the tap. So tap tap and MHC class 1 is joined by tapacin molecule. Okay, so that's the role of tapacin. What is the role of ERP57? So ERP57 actually is a oxidoreductase. So what it does is that it helps to break and form the disulfide bond between the MHC domains. Okay, so this forms the peptide loading complex. So what it does is that until the suitable peptide arrives, it holds the MHC molecule. Now once the proteins, so you can see that this is a ubiquitinated protein which is cleaved by the proteasome, and then it is transported by the tap, and here this peptide molecule will now be able to bind to the MHC molecule. Another molecule, ERAP, another protein, ERAP is also mentioned here. ERAP is an enzyme which is an aminopeptidase. So what it does is that if the peptide which is being transported into the ER lumen is longer than the MHC class 1 molecule can bind, so ERAP actually trims the protein. Okay, trims the end of the protein so that it can be of suitable length to bind to the MHC class 1 molecule. So now once this thing is finished, now what will happen, the all these chaperones, the calreticulin, ERP57, tapacin, this will dissociate from the MHC molecule, and then finally after the peptide binding, the MHC, the folding of the MHC will be actually finished. So until the peptide binds, the MHC molecule is not properly folded to be expressed, right? So once the peptide is bound, the folding is completed, and now this MHC molecule is being exported to the surface of the cell membrane. Okay, this is how the peptide transport takes place. Anyone having any question? Till this part?
Surasree, I asked that tap and tapacin, what is the difference? Like,
Tap is the transporter. So it is the transporter, it is ATP dependent. Yeah, role of ERP57 also I'm telling. Tap is the transporter. Okay, so what it does is that it transports the protein and the peptide fragments into the ER lumen from the cytosol. Okay, and tapacin is part of the peptide loading complex. So it creates a connection between the MHC class 2 MHC class 1 and the tap.
okay. And tap basically takes the protein from the cell and try to pull it up on the membrane.
From the cytosol, and
Yeah.
so actually what happens, this part I think you missed. This is the proteasome system. So here
Yeah.
what happens, these the proteins which are targeted to be degraded, they are added with ubiquitin molecules. They are polyubiquitinated, so many ubiquitin molecules are added to the protein, and then these proteins are they are actually now transported to the proteasome, where the proteins are degraded into the peptide fragments, and this process takes
Right.
place in the cytosol. Okay, so now the MHC molecule is present in the ER lumen. So how these peptide fragments will travel to the ER lumen? So for that, this transporter is the TAP.
Okay. Okay. Thanks.
And ERP57 is a thiol oxidoreductase. So what it does is that it helps to break and reform the disulfide linkage between the MHC domains, so that the MHC can be properly folded once it binds to the peptide. Is that okay? Okay. So now this is how it occurs in MHC class 1. Now, how is it different from MHC class 2? Now, I told you earlier also that MHC class 2 presents extracellular antigens, right? So they are not produced in the cytosol. The proteins are not produced in the cytosol. Neither will be they delivered by the TAP transporter to the ER lumen, right? So how does this take place? This is an overview of the antigen presentation by MHC class 2. And what happens here, you can see that since it is an extracellular antigen, the antigen is taken up by endocytosis, okay, into the cells, right? And in early endosomes, the pH is neutral. So here the endosomal proteases are inactive, but the proteases will be functional once the pH is acidic. So later on what happens, this acidification of the vesicle takes place, which activates the proteases, and the proteases now will degrade the antigenic protein to short peptide fragments. Alright, now after this acidification, after these proteins are fragmented, the MHC class 2 molecules will bind to the antigenic peptides, and then they will be expressed on the cell surface. So this is the overview of the event that is happening. I'd like to go into some detail here, which is there are certain terminologies that are going to come here. One is the invariant chain. The invariant chain. So the invariant chain is nothing but MHC class 2 associated invariant chain. Okay, what is it? It is a membrane glycoprotein, and its amino terminus resides in the cytosol, and its transmembrane region spans the ER lumen. Okay, alright. So what it does is that this is actually the chain which binds to the MHC class 2 molecules until and unless they can bind to the peptide. Okay, so when the MHC class 2 molecule is being produced in the endoplasmic reticulum, this invariant chain here, it remains bound to the class 2 molecule, and it protects or prevents the MHC class 2 molecule to bind to any peptides, alright, or any misfolded proteins. Now this will actually be cleaved off from the endoplasmic reticulum, and it now will travel in vesicle. So you know, from endoplasmic reticulum to Golgi to finally the endosomal pathway, right? So this is now here, it travels in the vesicle, and finally when the vesicle is acidified, so when the pH becomes acidic, the invariant chain, you can see here, invariant chain is cleaved off. Okay, so it is cleaved off in the proteases in the acidified endosome, and it there remains a short peptide which is still bound to the MHC class 2 molecule, which is the clip. Clip is nothing but class 2 associated invariant chain peptide. Okay, and here you can see clearly that the clip remains bound to the peptide binding groove of the MHC class 2 molecule, right? So clip is the fragment which is still bound to the MHC class 2 in the peptide binding groove, and you can understand that unless and until clip is removed, the MHC class 2 molecule will not be able to bind to the antigenic peptide, right? Now in the meanwhile, the endocytosis of the antigens take place, right? And they are degraded to peptides in the endosomes in a similar way, like the invariant chain was also degraded, and it, but the clip peptide is still bound to the MHC class 2, right? So therefore this these antigenic peptides cannot bind to the MHC class 2 molecule. Now another MHC class 2 like proteins are present in the cell which which will finally help in the binding of the antigenic peptide to the MHC. What are these? These are HLA-DM and HLA-DO. What is HLA-D? HLA is what? HLA is human leukocyte antigen. It is another name for MHC actually. So what happens? So unless this clip molecule is replaced, the antigen binding cannot take place. So here this HLA-DM helps here. So what it does is HLA-DM is not present on the cell surface, but it is found in these endosomal compartments here, in these endosomal vesicles. That also contains the clip and the MHC class 2 molecules, right? HLA-DM is present here, and what it does is that once the clip is actually dissociated, HLA-DM binds to the MHC class 2 and it stabilizes the empty MHC class 2 molecule. Okay, so HLA-DM stabilizes this empty MHC class 2 once the clip is dissociated, and once this is empty, therefore these antigenic peptides will now be able to bind to the antigen binding groove or the peptide binding cleft of the MHC molecule, right? So here this HLA-DM, the binding of the HLA-DM induces changes in the structure of the MHC class 2 molecule and holds this part of the peptide binding groove in a partially open configuration. So here you can see that this is kind of an open conformation, right? So binding of HLA-DM induces this structural change in the MHC class 2 molecule and it holds the peptide binding cleft in a partially open configuration, so that now the antigen peptides can now bind to the MHC class 2 molecule, and once this binding is completed, the MHC class 2 molecule will be properly folded and now it will be transported to the cell surface. Alright, another molecule that I mentioned here, which is the HLA-DO. HLA-DO is nothing but a negative regulator of HLA-DM. So this is also a class 2 MHC class 2 like molecule, okay, and it is produced in the thymic epithelial cells, the B cells and the dendritic cells, right? So this molecule is also not present in the cell surface, but it is present in the intracellular vesicles, like that of HLA-DM also. Okay, now how is it a negative regulator of HLA-DM? So when this DM-DO dimer, so HLA-DM and HLA-DO form a dimer together. So when this DM-DO dimer reaches the acidified endocytic compartment, okay, when this DM-DO dimer reaches this compartment, then what happens? HLA-DO is dissociated slowly. So HLA-DO is dissociated slowly which is why HLA-DM will now be able to catalyze peptide editing for the MHC class 2 molecule. Okay, so initially what happens, the HLA-DO and DM, HLA-DO and DM, they form a dimer together, right? And this dimer is present in the acidified endosomal compartment, but once this HLA-DO is dissociated, then the HLA-DM will be free and it can bind to the MHC class 2 molecule, so that now it can release the clip and it can now bind to the antigenic peptide and finally be transported to the cell surface. Okay, is that clear? Any doubts here? This is how the antigen presentation of MHC class 1 molecule and class 2 molecule are different. So in class 1 molecule, the binding of the antigenic peptide was happening to MHC in the ER lumen. However, in this case of MHC class 2, the binding is occurring in the acidified endosomal compartment, where there are multiple proteins that are involved. First is the invariant chain, which binds to the MHC class 2 and does not let it bind to the peptides prematurely. Then HLA-TM and HLA-TO, which help the MHC class 2 to bind to finally the antigenic peptide. Alright, if anyone having no question, then so these are the terminologies that are used in the previous slide: the invariant chain, clip, HLA-DM and HLA-TO. Now I'd like to go on to some questions here. Please try to answer them. So most of you are answering C, which is like the loading is regulated by both of them, the HLA-DM and HLA-DO. One is a positive regulator, another is a negative regulator. So the answer is C. You're correct. Okay, moving on to the next question. Sorry. Before moving on to the next question, let's come back here. One thing is that the peptide MHC complexes, they are continuously continuously recycled. The peptide MHC complexes which are present on the cell surface, they are continuously recycled from the surface, and what is done is that they are degraded in the cells by the proteasome, and then new MHC class 2 MHC molecules with the peptide MHC complexes will be now expressed on the cells. So there is continuous recycling which takes place. Now suppose a dendritic cell, here it is a dendritic cell which is shown here in this figure. Suppose a dendritic cell undergoes or it comes in contact with a pathogen. And once it comes in contact with the pathogen, it will engulf the pathogen, or there will be some kind of signaling by the PRRs, right, the pathogen recognition receptors. There will be some kind of signaling, and then they will be activated, right, and they will produce this the antigenic peptides. They will now express these antigenic peptides by the help of MHC molecules on their surface. right? Now, so this, if this happens during the encounter with the pathogen, now this peptide MHC complex is very essential for the body to be actually to be transported to the right place, so that the body can understand that this kind of infection is going on and we need to tackle it. Alright, so now you tell me, if the MHC class 2 molecules or the MHC molecules and the MHC peptide complex is now recycled after a few time, then obviously this information that there has been a pathogen attack which has been recognized by this dendritic cell, this information will be lost, right? So therefore there has to be a system so that once the dendritic cell like is um, gets contact with the pathogen, then there will be some system which will actually prevent the further MHC um, recycling from the surface. So this recycling has to be prevented, so that now this dendritic cell can now uh, travel to the lymph node, okay, to convey this information that it has come in contact with the pathogen to the naive T cells, right? So therefore this kind of traveling of the dendritic cells to the lymph node, this will take some time and it may take few hours. So in that period of time, this information that the MHC molecule presenting the antigenic peptide on the surface cannot be recycled. So this is actually controlled by one E3 ligase, which is March1. March1 is membrane associated ring finger E3 ligase. So what is that? First of all, what is E3 ligase? Can anyone tell me what is E3 ligase?
Ligase, I heard that it's join something. Like when we are seeing the recombination, there's some ligase enzymes. So
Yeah,
it's join something.
yes, very good. Join something, but here it joins ubiquitin. Okay. So this E3 ligase is actually part of the ubiquitin proteasome system. Okay. So there are many E3 ligases which are present in the cells, but this particular E3 ligase, so what it does is that it joins ubiquitin molecules to the target protein, so that the protein can be degraded, right? The protein can be taken to the proteasomal system. So this March1 E3 ligase, where is it present? It is present on the it is expressed in the B cells, the dendritic cells and the macrophages. Okay, so the antigen presenting cells, they have this March1 E3 ligase. And remember one thing, that they are constitutively present, constitutively expressed. So they are always expressed in these kind of cells. Or also they can also be induced, like their expression level can increase during any infection. Alright, now what it does is that it resides in the membrane of a recycling endosomal compartment. So here you can see, this is the endosomal compartment and this is the March1 ligase. So it resides in this, on the membrane of this kind of endosomal compartments, where it attaches, you can see, it attaches the ubiquitin molecules, so causes ubiquitination of the MHC molecules. Okay, so MHC molecules means what? Once the MHC molecules are taken in from the surface, from the cell surface, so they are for the recycling purpose. So now they will be actually targeted with this March1 E3 ligase, so that the March1 can now ubiquitinate the MHC class 2, and then because of this ubiquitination, this ligase, this MHC molecule will now be degraded by the proteasomal system. Alright, but what happens is that when there is an infection, this March1 E3 ligase pathway is shut down. So what do I mean by shutdown? So usually their expression is reduced, or the expression of the March1 ligase is blocked. So what happens here, you can see that this is a TLR, okay, and once it comes in contact with the pathogens or the pathogenic this LPS kind of lipopolysaccharides, right? These are some pathogenic signals, or the PAMPs, right? Pathogen associated molecular patterns. When this TLR will bind to this kind of PAMPs, this, so they will understand that there has been some infection, right? And then therefore what will happen, this TLR will signal now to block the expression from the March1 gene. So now the half-life of the March1 will be now reduced. So it will be, there will be very less March1 molecules present, because less expression of the molecules will take place. Now because of this because of this reduction in the lifetime of the March1, the lifetime of the MHC molecules will now increase. So you can see here the increase in the lifetime of the MHC molecules will take place. Okay, which means what? There will be no recycling that will take place. Okay, so now MHC molecules will now be accumulated on the cell surface, and they will present the peptides which is acquired during the time of this activation by the pathogen. Okay, so now this activated dendritic cell can accumulate these MHC class 2 antigenic peptide complexes on their cell surface, which is produced during the encounter of the pathogen, and then these molecules will now, these dendritic cells will now travel to the lymph nodes, where it will deliver this signal to the T cells. Also, this decrease in expression of the March1 also regulates CD86 expression. So if you remember what is CD86, CD86 is nothing but B7, right? CD86 or B7, which is another co-receptor. So it will also help in, so it will also increase the expression of this CD86 molecule, which is a co-stimulatory molecule, right? And once these dendritic cells reach the lymph nodes, they will be activated, and they will also have higher CD86 level, so that it provides the signal to the T cells for activation. Alright, so this is the transport of these activated T cells. Once it expresses the MHC class 2 molecules with the antigenic peptide, and then how it will be transported to the T cells, and what happens in between. So this importance of the role of E3 ligase March1. Any doubts?
Yeah, like I thought like some MHC will be always represented on the surface. So as here I understand, if there is no pathogen coming, so then there will not be any MHC on the surface.
No, not exactly like that. I told you in the last session also that there will be some the MHC molecules will also express some self antigens on its surface. So the MHCs are always expressing some peptides on the surface. But if that is not so when this kind of like any cell is expressing this MHC peptide complex, if that is not recognized by the T cells, then what will happen? So that means the T cell will recognize it as a self, and it will not react to it, right? It will not be activated. So then what will happen? These MHC molecules will be recycled. So they will, like, what will be their function if they are expressed on the cell surface without, like, if they are not pathogenic, right? So these MHC molecules are continuously recycled. That is why, because
So
all protein
this
molecules.
ubiquination will happen, like it will be tagged on the, like when it is on the surface, the MHC.
Now, once it is taken inside, then
Okay.
in these endosomal compartments, this March1 in E3 ligase will be present, which will ubiquitinate the MHC molecules, and then they will be taken for degradation.
Okay, so from the surface, it will, if it is not being used, then it will be taken into this
Endocytosis,
vesicles.
yes, yes,
Uh, okay, and another question: this protein, like I understand, like this MH will be degraded in the proteasome only, right?
yes.
And this ubiquitin, this protein, where it is generated in the protease or somewhere else?
No, the ubiquitin protein is actually very commonly available in the cell. So it is actually ubiquitously expressed. So which is why it is named as ubiquitin. Okay, so it is
Okay.
always expressed. So it is found in the like cytosol, and whenever there is a, so it is actually a degradation. So it is some agent which will degrade the proteins that are necessary to be degraded, right? So these ubiquitin proteins are associated. This is a very small protein. They are associated with any of the proteins which are to be degraded, right? And once these ubiquitin molecules, they are associated with the protein, once they bind to the protein is targeted for proteasomal degradation. Okay?
So it's just available in the cell, like it's
In the
expressed
cytosol, yes,
very abundantly.
yes, in the cytosol,
Okay.
right?
Okay.
So any doubt here? Any other doubt? If not, then try to answer this question. Take your time, go through these options carefully, and then choose the true statements. Okay, tell me if you think that any of these statements is wrong. Does anyone think statement number 1 is wrong?
Yes, ma'am. One is wrong.
Why? I'll tell you that it is correct
In
because
yes, ma'am. In lecture, you tell na.
they are expressed in B cells, dendritic cells, and macrophages. Okay. Acha, Sandeep is saying it is not. It does not reside in the membrane. But here, you carefully look at this picture. Where is this March 1 present? It is on the membrane of the endosomal compartment, of the recycling endosome, right?
Oh, I thought it's cytosol.
No, it's not cytosolic. It's on the membrane. So this is the transmembrane region. Okay, it's on the membrane, present on the membrane. All right, and the third option. So 1 is correct, 2 is also correct. The third option has to be correct, that it ubiquitinates the cytoplasmic tail of class 2 MHC molecules. So check here. This is the cytoplasmic tail, right? This is the transmembrane portion, and this is the cytoplasmic tail. Cytoplasmic tails means what? Something which is in the cytoplasm of the MHC class 2 molecules, right? So this tail is ubiquitinated by the MARCH1 ligase. Is that clear? All of you? So, therefore, all these options are correct. Okay. So, 1, 2 and 3 all are correct. Okay. And last topic for today, that is MHC restriction. So, this I mentioned previously also, that T cells will only recognize the antigenic peptide when it is presented by the suitable MHC molecules, right? So T cell can only recognize this antigenic peptide. This red one is the antigenic peptide here, and this is the MHC molecule, and this is the TCR, which can bind to the MHC molecule and simultaneously bind to the antigenic peptide, where it will recognize this antigenic peptide. Now, remember one thing, that TCR is also TCR can also recognize self versus non-self MHC. So this self versus non-self term I was taking previously also, where I was mentioning that the antigens, if it is a antigen of the person, like of the individual, like whether it is self antigen or whether it is non-self antigen, that has to be recognized by the immune system. Otherwise there will be cross reactivity against the self antigen, which is not required, which will be not harmful for the body. But this self versus non-self MHC recognition also occurs by the TCR. So what does it mean? TCR can recognize the MHC alleles or the MHC products which are expressed by one individual, whereas if there is a non-self MHC, so suppose I am an individual, there is another individual X. If the individual X's MHC molecules are transplanted in my body, okay, then my TCR or the T cells will recognize that MHC molecule as a non-self antigen. Okay, so the MHC molecule will be recognized as an antigen, and therefore it will be treated as an antigen only, right? So there will be immune responses generated against the MHC molecule, whereas this is only MHC, nothing else but MHC, not any pathogen, but MHC of another individual, okay, and if that is different from my MHC. So here you can see that this is MHCB. When it binds to the X peptide X, okay, the T cell, there is no recognition of the T cell. The TCR cannot recognize this other MHC bound antigenic peptide, okay, and this is something that is the intrinsic property of the TCR, like that is it can recognize the MHC molecules. However, and also one other thing, that is, if suppose my own MHC molecule binds to a peptide which it is not supposed to bind, then also TCR will not be able to recognize this MHC peptide complex. So the MHC molecule, the correct MHC molecule, has to bind to the correct antigen. This will actually determine the TCR recognition. So now this I was mentioning, that if there is a tissue transplantation, so the MHC molecules between two individuals are very different because of the polymorphism and the polygyny events, right? And this I was also discussing, that between the siblings also. There is a huge difference between the MHC molecules between the siblings. So if there is a transplantation, if there is a transplantation that has to be done, organ transplantation or tissue transplantation, then what happens? So you are taking this tissue from the from another individual, and this tissue will have MHC molecules. So the MHC molecules that are present in the tissue, right? And these MHC molecules, if they are different from my self MHC molecules, then what happens? These are my T cells will recognize that MHC molecule as non-self, and it will generate immune response against these non-self MHC molecules, which is why the grafts, the transplants are often rejected. Okay, this we have heard that often the grafts are rejected, the tissue transplantation does not work well, right? So for this transplantation to be proper, there has to be a MHC compatibility, right? So that the TCR cannot recognize the MHC, the TCR should not recognize any non-self MHC as, and it should not generate any immune response, otherwise the transplant might be rejected. So this kind of T cells which generate immune response against non-self MHC molecules are called alloreactive T cells. Okay, so if you have understood that, let's answer this question.
I just want to ask one thing. Like,
Yes.
if we are transplanting this some, let's say, liver, so can we not transfer the stem cells also to get the T cell to match with that MHC?
Yeah, but you know, like your body, the T cells which are present in your body, right? They are in much more abundance than the organ that you are actually um, transplanting, right? So these abundant T cells will, if they recognize that particular MHC as non-self, then this will cause the rejection of the transplant. Okay, however, there are several methods by which these transplants are now like there are methods by which you can improve this kind of transplantation event. And you know, right now there are transplants which are going on, right? Recently a heart transplant also took place. The patient was well for like 6 months or so, then suddenly the health of the patient deteriorated and he actually eventually passed away. But that was not because of the rejection of the transplant. So heart transplant, liver transplant, these are currently occurring in the medical science, the medical field, and there are several ways by which you can actually surpass or bypass these kind of events, these kind of alloreactivity. Okay, later on we will have you will have these lectures where these events will be discussed, this transplantation will be discussed.
Okay, okay.
The T cells, I mean to say the host. So the T cells of the host are much more in number than the T cells of the organ
No,
that you have.
I was thinking like if you inject the stem cells, then probably it will slowly proliferate, right? And then it will become also abundant.
These kind of things are actually done. You are kind of right. So there are other ways
Okay.
also where you can actually block this kind of alloreactivity. So there are some like some ways which we will discuss later on.
Okay.
Okay. So the answer to this is the alloreactive T cells, right? Okay. So that is all from my side for today. If you have any other doubts, please ask.
Ma'am,
Yes.
ma'am, exam date, application date, last date.
I'm Actually,
application date, ma'am.
I'm not very sure about that. Please check the course portal for that. I'm not very sure.
Okay, ma'am. One second, ma'am. Any Tamil persons, students? Hello?
Yes, ma'am.
கீர்த்தனா,
Yes.
தமிழ் தெரியுதுண்ணா உங்களுக்கு?
தெரியும்.
இப்போ மேம் கிட்ட எக்ஸாம் டேட் எப்போ லாஸ்ட் டேட் அப்ளை பண்ணணும், எப்படி அப்ளை பண்ணணும்னு கொஞ்சம் கேளுங்க மேம்.
லாஸ்ட் டேட் வந்து மேம்க்கு வந்து கரெக்டா தெரியலன்னு சொல்றாங்க.
ஓகே, இப்போ ஏதோ செப்டம்பர் 12ன்னு சொன்னாங்க இந்த கிளாஸ், வேற டாபிக் இதில். ஓகேங்களா, புரியுதா?
நீங்க வந்து ஜிமெயில பாருங்க, அதுல செப்டம்பர்